actual muscle fiber and start talking about something called a sarcomere. And a sarcomere is the smallest contractile unit of a muscle. And that's really very important for you guys to understand, because all of the muscle's contraction is happening at this very, very small level of the myofibril. And so many different myofibrils are contracting all at once in order for a muscle cell to contract and then the muscle to contract itself. A sarcomere is a region of a myofibril. Myofibrils are within the muscle fiber. So the muscle cell itself, the region between two Z discs or lines, you'll see Z disc or Z line, they mean the same thing. Each myofibril are composed of myofilaments, and the myofilaments are made up of protein. And again, I touched on this in the last lecture. We have many mitochondria in our muscle cells. But we also, in skeletal muscle, we have many nuclei. And if you guys can remember from biology that the nucleus houses DNA, and then DNA ultimately codes for every single one of your proteins. That's why we have so many nuclei in our skeletal muscles, because we have all of these protein fibers that need to be made in order for contraction to occur. There are two types of myofilaments. There's a thick and a thin. And in this picture, you're seeing one sarcomere. So here's a Z disc or a Z line. Here's the other Z disc or Z line. We have our thin myofilaments attached to the Z line. These are called actin filaments, and it's a protein. And then we have our thick myofilament called myosin here that is red. And when you're looking at this, because of the fact that we end up having thick and thin filaments either alone or overlapping, this is how we get those striations. In the I band, you can see that you only have the thin filaments, and so you're going to have a lighter coloration. And in the A band, you have both. You have your thick and your thin filaments. And so that is going to make it darker, and that causes those striations that you see in a skeletal muscle. So the banding pattern, this is where the myofilament banding pattern, this is where we get our striations. The thick filaments extend the entire length of an A band. The thin filaments extend across the I band and part way into the A band, and then the Z disc or Z line is another sheet of proteins. Then you have this other portion of the myofilament where the thin filaments do not overlap the thick filaments, and you have something called an H zone. And then you have M lines, which appear darker due to the presence of a different type of protein called desmin. So again, here is a picture, the same picture that I showed, but now we've extended it out and expanded it, I guess, out so that you can see it a little bit better. And so you have your Z disc, you have your thin actin, that's the purple, and then you have your thick myosin protein. And so we're going to, right here, if you're looking at this I band, you basically, it's going to be thinner because you have mostly actin. The A band, you have mostly myosin, and then you can see this thicker portion called the M line. That's just or a darker portion called the M line because it has a special protein. And then the H zone just has your thicker filament in it. So like I've been saying, the thick filaments are composed of a protein called myosin. And myosin are really unique looking proteins because they have a tail and two heads. So they're kind of the opposite of a phospholipid where it has one head and two tails. The tails are interwoven, so they're wrapped around one another. And the heads, they have these two little heads that are going to form something called a cross bridge, which is going to become important when we talk about the sliding filament theory. So this is what a myosin looks like. This is a protein, and you can see that along the myosin area, there's many different myosin molecules kind of interwoven, but the tail is going to be, tail will be wound together, and then you have these two heads that are eventually going to form the cross bridge. The thin filaments are composed of a protein called actin, and actin is going to have this little site on it, it's called an active site, where the myosin is going to attach during the sliding filament theory. And then there are two other proteins that are attached to the actin called tropomyosin and troponin. And once we get into the sliding filament theory, you'll understand what they do for the actual muscle contraction. And so this is what the thin filament looks like, and it is actin. So the purple balls are actin. The dark portion is the active site. The kind of orange strand is tropomyosin, and then the yellow is the troponin. So this is what it looks like on the sarcomere. So if you can picture, I had that picture up before, and it showed the Z line. So if you can picture that the Z line or Z disc would be here and here. Now you have your thick filament, that's your myosin. You've got your thin filament, that's your actin. And eventually there's going to be sliding of the two in order to contract a muscle. Now, along with those myofilaments and myofibrils, you have a new named organelle called sarcoplasmic reticulum. And sarcoplasmic reticulum is just a new name for smooth endoplasmic reticulum that's specific to muscle cells. And in the very first chapter, we learned this, but I'm sure you don't remember, that sarcoplasmic or smooth endoplasmic reticulum in muscle cells stores calcium. And that's really important because you need a lot of calcium for muscle contraction. And so along with this sarcoplasmic reticulum that kind of surrounds the muscle cell and the muscle fiber, you have something called terminal cisternae that's kind of woven in, and you're going to be storing and releasing calcium. And then you have something called elongated T-tubules. And what these tubules do are they are going to allow for the muscle contraction to get to every single fiber. And I will explain this a little bit more tomorrow. So this is what your muscle cell actually looks like. So right here we have those little myofibrils. We've got your myosin. We've got your actin, we've got all of those, bands that are allowing it to be striated, and then this blue portion is the sarcoplasmic reticulum. And so the sarcoplasmic reticulum is going to be housing calcium and releasing calcium when needed. And then on the sides here, you have something called the terminal cisternae, the two kind of blue rounded areas right here, and then this yellow portion is called the T-tubule. And the T-tubule, is, you can see, extends all the way through so that we can get the message throughout the entire muscle cell itself. So this is the last slide, and like I said, tomorrow I'll be explaining this a little bit more. But the T-tubules are continuous with the sarcolemma, which is the cell membrane. So they're going to go through the entire cell, and they allow us to conduct impulses to the deepest regions of the muscle. And that's really important because we need each myofibril and each myofilament to be contracting to contract the muscle overall. And they're going to signal for the release of calcium.